right now, like as you're listening to my voice, there are 2 meters of this microscopic, twisted ladder actively unzipping itself inside almost every single one of your trillions of cells, which is wild on its own, but here is the most shocking part about this frenetic little construction site inside your body.
It's the exact same machinery everywhere.
Exactly. It involves the exact same molecular ladder you would find if you zoomed in on the towering eucalypt tree swaying outside, or the microscopic bacterium invisible on your skin, or even like a mushroom growing on a fallen log.
Yeah, which is completely counterintuitive, right?
Right, because we look around at this overwhelming, just staggering variety of life on Earth, and I mean, it feels like common sense to assume that the blueprints for all these wildly different living things must be written in, well, completely different biological languages. But they aren't.
No, they really aren't. Every single living organism on this planet stores its biological instructions in the exact same molecule, written in the identical chemical alphabet.
And that brings us to our mission for this deep dive, because today we are going to thoroughly unpack this molecular manual of life, which is, of course, DNA. We're going straight into the physical mechanics of it, like what is this molecule physically made of?
Great, the actual architecture.
Yeah. How does it manage the physics of perfectly copying itself without just, snapping in half? How does it actually dictate whether you have brown eyes or maybe a risk for a certain disease? And what is modern science doing now that we can finally read this manual in real time?
It's a lot of ground to cover.
Yeah.
But it's so important.
It is. And I want to say to you, the listener, right up front, this isn't just some abstract textbook theory we're talking about today. This is the story of the literal biological machinery that is keeping you alive, maintaining your tissues, and dictating your biological reality right at this very second.
Yeah, grounding it in our own bodies is honestly the best way to understand it. Because when we fully grasp the mechanics of what is happening inside our own cells, we simultaneously unlock the biggest picture possible.
The universal picture.
Exactly. The universality of DNA. I mean, the fact that you and that mushroom and that eucalypt tree all operate on the exact same molecular code. It's the most profound, undeniable piece of evidence we have that everything alive today descends from a single common ancestor.
Because the odds of it happening twice are
basically zero. The statistical probability of this highly specific, incredibly complex chemical alphabet evolving independently multiple times in the exact same way is effectively zero. So you're looking at a shared heritage.
Okay, so if we share this underlying operating system with the rest of the natural world, I mean, we really need to know how it actually works. Let's zoom all the way down, past the organs, past the tissues, down into a single microscopic cell and into the nucleus right at its center. What are we physically looking at here? What is the actual architecture of life?
Well, let's start by just demystifying the name itself, because the name tells you exactly how it's built. DNA stands for deoxyribonucleic acid.
Deoxyribonucleic acid. It's a mouthful.
It is, but we can break that down. Deoxyribose. That refers to the specific type of sugar molecule that forms the foundation of its structure.
Wait, sugar? Like actual sugar?
Chemically speaking, yes. A type of sugar. Then the nucleic part simply tells us where we usually find it. It's safely housed inside the nucleus of the cell.
Okay, sugar in the nucleus. And the acid part.
The acid refers to the fact that under normal cellular conditions, the molecule has a tendency to give up a proton, which makes it chemically acidic.
So if we put it all together, we basically have an acidic sugar molecule sitting in the nucleus.
Essentially, yes.
But the shape of it is what everyone really recognizes, right? The double helix, that famous twisted ladder. But I really want to push on that for a second. Why is it twisted? Because we always draw it as a spiral, but molecules don't just choose a shape to look elegant for a textbook cover. There has to be a physical, chemical reason it twists like that.
Oh, there absolutely is.
Yeah.
The twist is entirely driven by the thermodynamics of the cell environment.
Thermodynamics.
Yeah. To understand that, we have to look at the ladder's 2 main components. You have the side rails and the rungs. So the 2 long side rails, the parts you would hold if you were, actually climbing it.
Right.
They're made as an alternating pattern of that deoxyribose sugar and a phosphate molecule. We call this the sugar-phosphate backbone.
Okay, so it's just sugar, phosphate, millions of times over.
Exactly. And crucially, this backbone carries a negative electrical charge, and it is hydrophilic.
Meaning it loves water.
Right. It readily interacts with the water that fills the cell. But the rungs of the ladder, however, the steps, are made of completely different molecules called bases. And these bases are hydrophobic. They strongly repel water.
Oh, wow. Okay, so you have a molecule where the outside edge desperately wants to be in water, and the inside rungs desperately hate water, and the whole thing is suspended in a cell that is basically mostly water.
You've hit the exact physical dilemma. The molecule naturally wants to minimize its energy state. So the water-loving backbone forms this protective outer shield, and the water-repelling bases stack up on the inside to hide from the water.
Like huddling together for warmth.
Yeah, exactly. But here's the catch. Because of the specific angles of the chemical bonds between the sugar and those bases, the only physical way for those bases to stack tightly enough to completely lock out the water is if the entire structure twists into a spiral.
No way. So the cell's watery environment literally forces the ladder into that iconic double helix shape.
It does. The environment squeezes it into a spiral staircase.
That makes so much more sense than just assuming it's some random arbitrary design. It's a thermodynamic inevitability.
Okay, so this sugar-phosphate backbone is essentially structural scaffolding. It gives the molecule its shape and stability, it protects the inside from water, but it isn't the actual information, right? It's just the physical track holding the data.
Yes. Think of it like a highly advanced, ultra-durable magnetic tape.
Like an old VHS or a cassette tape?
Mm.
Yeah, perfect. The plastic casing of the tape is absolutely vital for protecting the data from degrading, but you don't read the plastic casing to watch the movie. You read the magnetic variations on the inside tape.
Mm.
In DNA, the information in the movie is written entirely in those hydrophobic rungs hidden away in the center of the helix.
And those rungs are where we encounter the alphabet. Because each rung is made of two chemical units that meet in the middle, right? These are the bases. And I feel like here is where the sheer simplicity of this system becomes almost hard to believe.
It really is. Because there are only 4 bases in this entire system, just 4. Adenine, thymine, guanine, and cytosine.
Which are usually just abbreviated to A, T, G, and C.
Right, A, T, G, C.
4 letters. I mean, we use an alphabet of 26 letters to write a simple email. And even then, we struggle to articulate complex ideas sometimes. But nature uses precisely 4 chemical letters to write the instructions for, like the human brain or the bioluminescence of a firefly, the venom of a cobra, the bark of a tree, everything. It all comes down to just the sequence of those 4 letters along the immense length of that twisted ladder.
It's an absolute masterclass in efficiency. But, it poses a massive mechanical problem.
What do you mean?
Well, if this fragile, twisted ladder holds the complete memory of a species, how does it physically copy itself without losing information? I mean, when a cell divides to make a new cell, which is happening literally millions of times a second in our bodies, how does it duplicate billions of letters perfectly?
Right, because if you gave me a book with 3 billion letters in it and told me to hand copy it, I would make thousands of typos, probably on the first page. How does a mindless chemical structure avoid losing the plot?
The answer to that lies in the single most important structural rule of DNA. This rule dictates exactly how those 4 letters interact with each other to form the rungs, because they don't just pair up randomly. There's a strict physical pairing rule. A always pairs with T, and G always pairs with C.
A to T, G to C. Okay, but again, I have to ask why. Because we can't just memorize the rule for this deep dive. We need to know the mechanism. Why can't an A pair with a G?
It comes down to their physical size and their chemical bonding points. So the 4 bases belong to 2 different chemical families. A and G are larger molecules. They have this double ring structure, and we call them purines. T and C are smaller, single ring molecules called pyrimidines.
So one is wide and one is narrow?
Yes. Now remember the 2 rails of the DNA backbone? They are kept at a perfectly uniform distance apart all the way down. If 2 large bases, like an A and a G, tried to pair together, the rung would be too wide and would visibly bulge the ladder outward.
Like a warped step on a staircase.
Exactly. And if 2 small bases paired, the rung would be too short and the ladder would pinch inward. So to keep the distance perfectly uniform, every single rung must consist of 1 large base and 1 small base.
Okay, so that explains why a large A has to pair with a small T or C, but why specifically T? Why not C? Because they're both small, right?
They are, but that specific pairing is dictated by hydrogen bonds. Think of hydrogen bonds like molecular Velcro. Adenine and thymine are chemically shaped so that they form exactly 2 hydrogen bonds with each other. Guanine and cytosine are shaped to form exactly 3 hydrogen bonds.
So that's a lock and key mechanism.
It really is.
So A and T are like a 2-prong electrical plug and a 2-prong outlet, and G and C are a 3-prong plug and a 3-prong outlet, and A physically cannot bond with the C because the prongs just don't line up.
Precisely. The physics of the molecules makes it impossible for them to pair incorrectly without causing severe physical distortion. And if that happens, the cell's proofreading machinery would immediately recognize it as an error and try to fix it. But this strict two-prong, three-prong pairing rule, ATGC, provides the ultimate built-in backup system.
How so?
Because if you know the sequence of letters on one side of the ladder, you inherently know the exact sequence on the opposing side.
Let's visualize that for the listener. So if I'm reading down one half of the ladder, right, just the left rail, and I see the sequence A, G, G, C, I know with mathematical certainty what is on the other side.
You do.
Because an A requires a T, AG requires a C. So an AG-TTC strand simply must be locked into a TCC-AG strand on the other side. The molecule essentially carries a mirrored backup copy of itself at all times.
Yeah, and that mirroring that specific realization was the foundational breakthrough made in 1953 by James Watson and Francis Crick. Though it is extremely crucial to note here that their model was heavily dependent on X-ray crystallography data.
Right, the famous photo 51.
Yes, produced by Rosalind Franklin. Her X-ray data proved the physical dimensions of the helix. And knowing the exact width of the helix is what allowed Watson and Crick to realize that the bases had to pair in this specific ATGC manner to fit inside.
And when they finally published their paper on this, they included what has to be just the most understated sentence in scientific history.
Oh, I know the one you mean.
Right. They casually wrote that this specific pairing, and I'm quoting here, immediately suggests a possible copying mechanism for the genetic material. Like, they essentially found the mechanical secret to all biological reproduction and just casually floated it as a possible mechanism at the end of the paper.
Well, they understood the gravity of it perfectly. They knew that if the two strands complement each other perfectly, like a mirror, you have a literal physical way to replicate the entire molecule.
So let's talk through that replication process. Because we often hear the analogy of a zipper on a jacket. You pull the zipper straight down the middle, separating the two halves. The teeth pulling apart are those hydrogen bonds, you know, the two-prong and three-prong connections we just talked about breaking. So you end up with two separate strands with all those half rungs exposed.
It's a great visual. But let's look at the actual molecular machinery doing the unzipping, because it doesn't just pull apart on its own by magic. An enzyme, which is a specialized protein worker called helicase, acts as the zipper pull.
Helicase.
Right. Helicase physically wedges itself between the two strands and just plows forward, breaking the hydrogen bonds at a staggering rate of up to 1,000 base pairs per second.
Wait, 1,000 per second?
Up to 1,000, yes.
But wait, if you have a piece of string that is tightly twisted into a spiral and you violently pull the two strands apart in the middle, the twisting tension gets pushed further down the string. Like if you've ever played with the twisted yarn, it gets tighter and tighter until the string just snarls into a knot or snaps entirely.
Yes. And DNA faces that exact physical threat. If helicase just kept unwinding at that speed, the tension ahead of the split would physically shatter the DNA backbone. The molecule would be destroyed.
So how does it survive its own unzipping?
The cell deploys another enzyme called topoisomerase.
Topoisomerase.
Yes. This enzyme travels ahead of the unzipping helicase. It temporarily snips the DNA backbone, literally cutting it, letting the tension spin out and relax, and then taping it back together in a fraction of a second.
That is insane. So we basically have a demolition crew plowing through the center of the building and an engineer running ahead to temporarily cut the structural beams to relieve the tension so the whole building doesn't collapse on itself.
That's a fantastic way to picture it. And once those strands are safely separated by helicase, they're exposed to the interior fluid of the nucleus. And that fluid is just flooded with free-floating individual bases, loose A's, T's, G's, and C's drifting around. This is where a third enzyme called DNA polymerase comes in.
The builder enzyme.
The builder. So DNA polymerase travels along the exposed single strand. And thanks to our strict pairing rule, it doesn't even have to, think about what to build. If it encounters an exposed A on the original strand, the physical shape of that A will only allow a free-floating T to click into place.
Because of the hydrogen bonds, the two-prong plug automatically finds the two-prong outlet. So as the builder moves down the separated strands, it just pulls these loose pieces out of the cellular soup and locks them in. And because it's happening on both of the separated halves simultaneously, you end up with two complete double helixes.
Where there was originally one double helix, there are now two perfectly identical copies.
Yeah.
And notice the elegance of the system. Each of the new double helices consists of one original parent strand and one freshly built daughter strand. Half is conserved from the original, and half is brand new.
Which guarantees that the information is perfectly preserved through generations and generations of cell division. And to ground this again for you listening right now, this orchestrated chaos, the helicase plowing, the topoisomerase snipping, the polymerase building at 1,000 letters a second is happening inside you right now.
Right, it's very second.
If you are recovering from a workout or fighting off a cold or honestly just maintaining the lining of your stomach, your body is executing this exact copying mechanism on billions of cells as you listen to this.
Which naturally leads us to a pretty staggering logistical issue regarding physical space.
Oh, absolutely. Because if we have billions of cells constantly doing this unzipping and copying, the sheer volume of this physical material must be absolutely enormous. I mean, how much DNA is actually inside us?
Well, let's isolate just one single microscopic human cell first. If you were to extract the DNA from the nucleus of one human cell and stretch it out into a single straight line, it would measure roughly 2 meters long.
2 meters, over 6 feet of DNA, inside one single completely invisible cell.
Yes. And the compartment it lives in, the nucleus, is only about 6 micrometers across. That is a few thousandths of a millimeter.
The geometry of that feels impossible. I'm trying to picture that, and if you scale that up, that's like trying to pack 24 miles of extremely fine fishing line into a single tennis ball. How does the cell prevent this massive thread from just becoming a hopelessly tangled knot? Because if it tangles, the helicase can't unzip it, right? The cell can't divide and the organism dies.
Exactly. The solution is an incredibly sophisticated packing mechanism. The cell doesn't just stuff the DNA inside randomly, it actively spools it. The long thread of the double helix wraps tightly around clusters of specialized proteins called histones.
Histones. And I remember reading they're positively charged, right? Why is that important?
Well, remember how we established earlier that the sugar phosphate backbone of the DNA has a negative charge?
Oh, opposite charges attract. The negative DNA physically snaps onto the positive histone proteins like magnets. It's an electrostatic hold.
Exactly. It's a very tight grip. The DNA wraps around a cluster of 8 histones, creating a structure called the nucleosome. If you looked at it under a powerful electron microscope at this specific stage, it looks exactly like beads on a string.
Okay, beads on a string, but that's still pretty long.
It is, so it doesn't stop there. Those beaded strings then coil up into a thicker fiber, and that fiber then supercoils, literally looping and folding back on itself again and again and again to create highly dense, highly organized structures.
Chromosomes.
Yes, a chromosome is simply this 2-meter-long thread of DNA wound around millions of these microscopic magnetic spools and coiled upon itself until it is dense enough to be moved around during cell division without tangling. And humans package our 2 meters of DNA into 46 distinct chromosomes, which come in 23 pairs. You inherit 23 from your biological mother and 23 from your biological father.
I want to run a quick thought experiment here to really hammer home the scale of this natural engineering for anyone listening. We know one single cell contains 2 meters of DNA, and an average adult human body contains roughly 30 trillion cells.
That's the generally accepted estimate, yes.
Yeah.
Give or take a few trillion.
Right. So if you somehow manage to magically extract the DNA from every single cell in your own body, uncoiled it from all those magnetic histone spools, and laid every single strand end to end in a straight line, how far would that physical thread reach?
The math requires astronomical distances. I mean, it wouldn't just stretch across the planet. If you laid all the DNA in your body end to end, it would stretch from the Earth to the sun and back again, roughly 60 times.
From the Earth to the sun, over 90 million miles, and back dozens of times. All of that is physically packed inside the boundaries of your skin.
It is a profound demonstration of how incredibly thin the DNA molecule actually is and how brilliantly the cell manages to organize its physical environment.
It is genuinely awe-inspiring. So we now understand the architecture. We know it's a twisted ladder made of 4 letters, locked together by hydrogen bonds, spooled around charged proteins, and we know how it unzips to perfectly copy itself. But, an instruction manual is completely useless unless someone is actually reading it to build something. What is this code actually doing all day to keep us alive?
Now we move from the structure of DNA to its active function. The primary job of most functional DNA sequences is to specify the creation of proteins.
And we really need to clarify what we mean by protein, because the everyday use of that word usually refers to, dietary nutrition. We think of a chicken breast or protein shakes at the gym.
Right, dietary protein is what we consume to get raw materials. But in molecular biology, proteins are the actual machinery of life. They are the physical agents that do all the work inside your cells.
So if a human cell is a bustling factory, the proteins aren't just the raw materials sitting on pallets. The proteins are the assembly line workers, the conveyor belts, the support beams holding up the roof, and the security guards checking IDs at the door.
That is a perfect analogy. Proteins are incredibly diverse. They are the enzymes that physically break down your food in your stomach. They form the structural fibers that give your body its physical shape, like the collagen holding your skin together and the keratin making up your hair and nails.
They are the signaling molecules too, right?
Yes, hormones like insulin are actually proteins that travel through your blood to tell cells to absorb sugar. Antibodies are highly specialized proteins that your immune system deploys to physically bind to and neutralize invading viruses. Hemoglobin is a protein complex inside your red blood cells, shaped specifically to trap oxygen in the lungs and release it in the tissues. Truly, every single biological action is carried out by a protein.
So the DNA isn't doing the work itself. The DNA is just the master blueprint sitting safely in the nucleus manager's office, dictating exactly how to build every single protein worker in the factory.
Precisely. But this creates a huge logistical barrier for the cell. The DNA is far too precious, and honestly too enormous, to ever leave the protective vault of the nucleus. If it goes out onto the messy, highly active factory floor of the main cell, it could easily be damaged or destroyed. So the cell uses a two-step communication system to get the instructions out safely.
It's kind of like having a priceless ancient reference text in a restricted library. You definitely can't take the book out to the workshop where there's grease and sparks. So step one has to be making a copy of just the specific page you need.
Exactly. We call step one transcription. When the cell needs to build a specific protein, say, it needs more insulin, an enzyme goes into the nucleus, locates the specific sequence of DNA containing the insulin recipe, unzips just that tiny section, and creates a working copy.
But it doesn't make a DNA copy, does it?
No, it transcribes the information into a slightly different, closely related molecule called messenger RNA, or mRNA.
Oh, mRNA, a term that became globally recognized during the development of the COVID-19 vaccines. How is RNA physically different from DNA?
Chemically, it uses a slightly different sugar for its backbone ribose instead of deoxyribose. And instead of the base thymine, the T, RNA uses a base called uracil, or U. Structurally, it is normally just a single strand, not a double helix.
Why the change in materials? Why not just use the exact same letters and make a mini DNA strand?
Because mRNA is inherently designed to be temporary. The cell doesn't want the factory floor permanently cluttered with old instructions. The chemical structure of RNA makes it far less stable than DNA. It degrades relatively quickly. It exists just long enough to deliver the message to the factory, and then it breaks down so the components can be recycled.
So we have transcribed our temporary mRNA copy, and because it's small and totally expendable, it is allowed to exit the nucleus vault and travel out into the main body of the cell. What is step two?
Step two is translation. The nucleotide language of the mRNA needs to be literally translated into the amino acid language of proteins. This translation is performed by a massive, complex molecular machine called a ribosome.
So the ribosome is the foreman on the factory floor reading the blueprint page. How does it physically read those chemical letters?
The ribosome basically clamps around the mRNA strand and begins reading the sequence, but it doesn't read one letter at a time. It reads the letters in discrete groups of 3. Each 3-letter sequence is called a codon.
Okay, so if the mRNA sequence is AUGCCA, the ribosome reads AUG as one distinct word and CCA as the next distinct word.
Exactly. And the magic of the genetic code is that each specific 3-letter codon corresponds to exactly one amino acid.
And amino acids are the physical building blocks that make up a protein.
Yes. Think of amino acids like biological Lego bricks. Life on Earth utilizes 20 different types of these amino acids. As the ribosome reads the mRNA, another molecule called transfer RNA, or tRNA, floats by. One end of the tRNA perfectly matches the 3-letter codon being read, and the other end carries the specific amino acid that codon asks for.
So the tRNA is a delivery driver.
It absolutely is. The ribosome reads the AUG codon, the corresponding tRNA delivery driver docks into place, and the ribosome clicks that amino acid onto the chain. Then the ribosome moves forward exactly 3 letters, reads the next codon, the next delivery driver arrives, and the ribosome clicks the new amino acid onto the first one.
This builds a long physical chain of amino acids, assembled in the precise order originally dictated by the DNA back in the nucleus.
And once the ribosome reaches a specific stop codon, it releases the chain. Now, because of the distinct chemical properties of the different amino acids in the chain, some love water, some hate it, some have positive charges, some have negative charges, the long chain instantly folds up on itself into a highly specific, complex 3D shape. And that folded shape is the functional protein ready to do its job.
Form dictates function. The sequence dictates the folding, and the folding dictates what the protein can actually do.
Exactly.
But let's pause and look at the mathematical logic of this three-letter codon system for a second, because there is an incredible protective mechanism hidden in the math here. We have an alphabet of 4 letters, and they are read in groups of 3. If you multiply that out, 4 times 4 times 4, there are exactly 64 possible combinations, or codons.
Correct. 64 possible 3-letter words.
But you just said the factory only uses 20 amino acid Lego bricks. If there are 64 words, but only 20 actual ingredients to ask for, what are the other 44 words doing?
They provide redundancy. Because there are 64 words and only 20 amino acids, most amino acids are coded for by multiple different codons. For example, the amino acid proline is requested by the codons CCU, CCC, CCA, and CCG. Any of those 4 sequences tells the ribosome, put a proline here.
There are chemical synonyms, multiple ways to say the exact same thing.
Exactly. The genetic code is highly degenerate, meaning there is built-in redundancy, particularly in the 3rd letter of the codon. And this redundancy is an evolutionary shield against chaos. Which brings us perfectly into what happens when the system actually makes a mistake.
Right, glitches in the matrix. Because if helicase and polymerase are rushing along, unzipping and copying billions of letters at lightning speed, mistakes are physically inevitable. What is a mutation, fundamentally?
Stripped of all the science fiction tropes from movies, a mutation is simply a change in the DNA sequence. A chemical typo. An A accidentally gets copied as a G, or maybe a letter gets skipped entirely.
How does that happen, though, beyond just polymerase making a sloppy typo during replication?
Well, the environment constantly damages our DNA. Ultraviolet light from the sun is a major culprit. When UV photons hit your skin cells, the physical energy can cause 2 adjacent thymine bases, 2 T's on the DNA strand, to physically fuse together into a knot called a thymine dimer, which the copying machinery really struggles to read.
Wow.
Yeah, and certain chemicals, radiation, and even the normal metabolic byproducts of simply converting your food to energy can physically alter the DNA structure inside your cells.
And when the copying machine hits that damage, it basically has to guess the letter, and a mutation is cemented into the code. Now, the cultural perception of a mutation is that it is inherently monstrous. It causes disease or radiation sickness or turns you into a superhero. But that's where the redundancy of the 64 codons comes in to save us, right?
Exactly. A huge percentage of point mutations, which is where a single letter is swapped, are entirely silent. Let's go back to our proline example. The original DNA says CCA, so the ribosome clicks a proline onto the chain. If a UV photon damages that DNA, and during repair, it mutates. So the sequence now reads CCG.
CCG is a synonym. It still codes for proline.
Precisely. The ribosome reads the mutated sequence, but the tRNA delivery driver still brings a proline. The resulting amino acid chain is 100% identical. The protein folds the exact same way and it functions perfectly. The mutation occurred, but it was physically meaningless. Furthermore, massive stretches of our DNA do not code for proteins at all. If a typo lands in certain non-coding regions, it often has absolutely no effect on the factory floor.
It's like a typo occurring in the blank margins of the manual. Nobody really cares. But we can't pretend all typos are harmless. When a typo occurs in a critical area, the physical consequences can be absolutely devastating.
It certainly can. If a single letter change occurs early in a critical gene, it might specify a completely different amino acid. If you swap a water-loving amino acid for a water-hating one, the entire long chain will fold incorrectly, and a misfolded protein is usually a completely broken machine.
Our source material gives a stark, really powerful example of this, sickle cell disease. Walk us through the physical mechanics of that single typo.
Right, so sickle cell disease is caused by a mutation in the gene that builds hemoglobin, which is the oxygen-carrying protein in your red blood cells. The original healthy sequence contains a codon that asks for an amino acid called glutamic acid, which is hydrophilic. It likes water.
Okay, so it helps the hemoglobin dissolve nicely in the fluid of the blood cell.
Exactly. But in sickle cell, a single DNA letter is swapped. Just one. An A is replaced by a T. That single change shifts the codon so that it now asks for valine instead of glutamic acid. And valine is hydrophobic. It strongly hates water.
Oh no. So suddenly on the outside of this protein, there is a physical patch that is desperately trying to hide from the watery environment of the cell.
Yes. And the only way the valine patch can hide from the water is by sticking to a hydrophobic valine patch on another mutated hemoglobin molecule nearby. The proteins begin to click together, forming long, rigid physical chains inside the red blood cell. These rigid chains stretch the cell out of its normal, flexible, round shape into a stiff, curved sickle shape.
And those stiff, sickle-shaped cells get physically stuck in the tiny capillaries of the blood vessels, causing severe pain and organ damage. All because one single A became a T, causing one water-loving building block to be swapped for a water-hating one.
It perfectly illustrates how incredibly sensitive the physical realities of our bodies are to the molecular realities of our DNA.
But this introduces a really profound philosophical and biological paradox. We are talking about these mutations, these typos, as the mechanism of devastating life-altering diseases. Yet these exact same copying errors are simultaneously the reason we exist at all.
That is the ultimate dual nature of DNA. While most mutations are silent, and some are disastrously harmful, a tiny, tiny fraction of mutations happen to alter a protein in a way that provides a physical advantage in a specific environment.
A typo that actually improves the machine.
Right. A mutation might slightly alter the shape of an enzyme, so it breaks down a new type of food source more efficiently. Or it might alter a pigment protein, providing better camouflage against predators. If that advantageous mutation helps the organism survive and reproduce, that specific typo gets passed on to the next generation.
This is natural selection in action.
It is the raw physical material of evolution. Natural selection is simply a filter. It can only select from the variations that exist in front of it. And mutation is the solitary source of that new variation. If DNA polymerase never made a typo, if DNA was perfectly, rigidly immune to environmental damage, life would be a stagnant loop. We would still be identical single-celled organisms in the ocean.
So the engine of genetic disease is the exact same mechanism as the engine of evolutionary diversity. Without the vulnerability that causes sickle cell or cancer, we wouldn't have the adaptability that gave us the human brain or the wings of an eagle or the bark on that tree.
They are completely inseparable realities of the exact same chemical mechanism.
Man, that's heavy. I want to shift our focus now to how we actively talk about this code in our everyday lives, because out in the world, people rarely talk about nucleotides or codons. We talk about genes, and this is an area where popular language causes massive misunderstandings about how our biology actually works.
Oh, definitely. The word gene carries a lot of cultural baggage. In molecular biology, a gene is generally defined as a specific sequence of DNA that contains the complete instructions to transcribe an RNA molecule, usually to build a specific protein.
So a gene is essentially one complete, discrete recipe in the larger cookbook.
That is a highly functional way to think about it. And understanding how many recipes we have leads to one of the most humbling discoveries in modern science. In the 1990s and early 2000s, when the International Human Genome Project was working to sequence human DNA for the first time, literally reading all 3 billion letters, there was intense speculation about how many genes we would actually find.
Which makes sense. I mean, humans are staggeringly complex. We have intricate immune systems, we have the cerebral cortex, we build civilizations. The assumption was that our cookbook must be incredibly sick.
The prevailing estimates guessed humans had around 100,000 to maybe 150,000 distinct genes. But when the sequencing was completed, the actual number revealed what became known as the roundworm dilemma.
Lay the numbers on us. Prepare to humble everyone listening.
It turns out humans possess roughly 20,000 protein-coding genes.
Only 20,000.
Yes. To put that in perspective, a microscopic transparent soil nematode, a tiny roundworm called C. elegans, which consists of exactly 959 cells total, has about 20,000 genes as well.
I think anyone listening right now is completely justified in feeling a little insulted by that. How is it physically possible that a complex human being with a brain capable of understanding its own existence operates on the exact same number of instruction manuals as a microscopic worm living in the dirt?
It forces us to realize that biological complexity is not simply a matter of gene count. It's not about how many recipes you own. It's about the sophistication of how and when you cook them. The answer to human complexity lies in gene expression and regulation.
Meaning, just because you possess a gene doesn't mean the factory's actually using it right now.
Precisely. Let's look at your own body again. Almost every single one of your 30 trillion cells contains the exact same 2 meters of DNA. The volt in your liver cell contains the exact same 20,000 genes as the volt in a neuron in your brain. Yet a liver cell is a blocky chemical filter processing toxins, and a neuron is a long, spindly wire firing electrical impulses.
They look and behave like they belong to completely different species. How do they differentiate if they have the same manual?
Because of regulation. In the liver cell, the genes responsible for building neural pathways are heavily repressed. They're physically locked down and switched off. The genes for processing toxins are wide open and highly active. In the neuron, the exact opposite is true. The complexity comes from the dynamic network of turning genes on, turning them off, or dialing them up and down like a dimmer switch.
How does the cell physically flip these switches? You mentioned earlier that huge portions of our DNA do not code for proteins. For a long time, the public was taught that this was just junk DNA.
Junk DNA is a term that biologists deeply, deeply regret. It was coined simply because we didn't initially understand what it did, as it didn't code for proteins. But we now know that this massive expanse of non-coding DNA is actually the control room.
It's the regulatory network.
Yes. These non-coding regions contain sequences called promoters and enhancers. They act as physical landing pads for specialized proteins called transcription factors. When a transcription factor binds to an enhancer region, it physically bends the DNA strand, bringing it into contact with a specific gene to initiate transcription. Furthermore, we have a whole layer of physical control called epigenetics.
Epigenetics, the physical modification of the DNA structure itself without changing the actual letters.
Exactly. The cell can attach a small chemical tag, a methyl group, directly to the cytosine bases in the DNA. These methyl tags physically prevent the transcription machinery from latching on, effectively silencing the gene. Or the cell can chemically modify those histone spools we talked about earlier. If the cell attaches an acetyl group to the histones, the spools relax their magnetic grip on the DNA, unwinding it slightly so the reading machinery can access it.
So the DNA isn't just a static string of code sitting there passively. It is constantly being physically folded, tagged, hidden, and exposed in response to the environment. The sheer level of control is mind-bending. And knowing how tightly regulated this is brings us perfectly to busting one of the most irritating tropes in popular science media.
Ah, the gene for myth.
Yes. You see a headline that says, "Scientists discover the gene for height," or "the gene for intelligence," or "a gene for aggressive behavior." It's everywhere.
It is a fundamentally flawed way of communicating genetics. It treats genes like singular on-off switches for incredibly complex physical and behavioral outcomes. Now, to be clear, single gene conditions do exist. We discussed sickle cell, where one typo in one gene definitively causes the disease. Huntington's disease and cystic fibrosis are other examples. But these are rare exceptions to the overall rule of biology.
For the vast majority of what makes you who you are, you can't point to a single spot on the twisted ladder and say, that's the tall gene.
You cannot. Most human traits, height, intelligence, risk for heart disease, susceptibility to depression, are polygenic. Poly meaning many. These traits are the culmination of hundreds, sometimes thousands, of different genetic variants scattered across your entire genome. Each individual variant might only contribute a millimeter to your height or a microscopic fraction of a percent to your disease risk.
It's a massive collective chorus of genes, not a single soloist.
Yeah.
And even then, the DNA isn't the final word because it interacts with the physical environment, right?
Environmental interaction is half the equation. Let's stick with height. You could inherit a polygenic profile that gives you the biological potential to be 6 feet tall. But if you suffer from severe malnutrition during childhood or a chronic illness that diverts your cellular energy away from bone growth, your environment will physically restrict your growth. You will not reach 6 feet, regardless of what the manual says.
The environment intercepts the instructions, which brings us to a quote from the source material that serves as a profound anchor for anyone trying to understand their own biology.
Oh, genes load the dice. They rarely determine the throw.
That is brilliant. The sequence of your A's, T's, G's, and C's establishes a set of probabilities. It sets the boundaries of your biological potential. It loads the dice. But how those dice actually roll, how your life literally manifests, is determined by the unimaginably complex interaction of those 20,000 genes being regulated by epigenetics, constantly responding to your diet, your stress levels, your environment, and sheer chance.
It is the perfect synthesis of genetics and environment. We are not rigid biological robots executing a static code. We are highly dynamic systems.
Okay, as we enter the final segment of our deep dive today, we need to talk about where this science is right now. Because we've gone from Watson and Crick drawing models on paper in the 1950s to the massive Human Genome Project in the 2000s. And the acceleration of this technology from abstract theory to routine, life-altering medical application is breathtaking.
The technological leap in genomic science over the last 2 decades is arguably the most rapid acceleration of capability in scientific history. Just look at the physical process of sequencing, at actually reading the A's, T's, G's, and C's. When the Human Genome Project began, they were using Sanger sequencing, which was incredibly slow and required immense physical infrastructure.
It was a monumental international effort. To sequence the very first complete human genome took 13 years of continuous work, and it cost nearly $3 billion.
13 years and billions of dollars to read the manual once. Fast forward to today. The advent of next-generation sequencing relies on massively parallel processing. Instead of reading the DNA strand end-to-end, machines shatter the DNA into millions of tiny fragments, read them all simultaneously on microscopic flow cells, and use immense computing power to stitch the sequence back together.
And what does that mean for the time and cost today?
Today, you can sequence a complete human genome in roughly 24 hours, and it costs a few $100s.
From $3 billion to $300. From 13 years to a single afternoon. When the cost of reading the blueprint drops that exponentially, it completely democratizes the science. It moves out of elite government research facilities and goes straight into the diagnostic labs of your local hospital.
And the real-world impact detailed in our source material is transforming medicine. In the realm of rare genetic diseases, what used to require a diagnostic odyssey, years of painful, invasive, and often incorrect testing, can now be solved by simply sequencing a child's genome and directly locating the single typographical error causing the condition.
It provides immediate answers. And it's completely revolutionizing oncology, too. Cancer, at its core, is a disease of mutated DNA.
Exactly. Cancer occurs when the genes regulating cell division mutate and get stuck in the on position. Today, oncologists routinely extract DNA from a patient's tumor and sequence it. They aren't just guessing which chemotherapy might work. They identify the exact molecular mutation driving that specific tumor and prescribe targeted drugs designed to chemically inhibit that exact mutant protein. It is precision medicine at the molecular level.
And it extends to public health, which we all experienced recently. When a new pathogen emerges, we no longer have to wait months to figure out what it is. Scientists sequence the genome of a new virus within days. We can watch the virus mutate in real time, tracking the specific sequence changes to see how it moves across borders.
Genomic surveillance is now the bedrock of epidemiology. And looking backward, this technology is rewriting our understanding of human history. Paleogenetics allows us to extract highly degraded DNA fragments from the bones of ancient hominins that have been buried in caves for tens of thousands of years.
We are reading the manuals of species that don't even exist anymore.
And by comparing those ancient sequences to modern human DNA, we have physically mapped out the migration routes of our ancestors. We even discovered that early humans successfully interbred with Neanderthals because the exact DNA sequences inherited from those ancient encounters are still physically present in the genomes of billions of people alive today.
The manual holds the history of our species, but the source material notes that we are already crossing the next great threshold. Because we have mastered reading the code, we can find the typos, but now we have the tools to actively rewrite it.
The development of gene editing technologies, most notably CRISPR-Cas9, has fundamentally changed our relationship with biology. CRISPR is essentially a bacterial immune system that scientists have repurposed into programmable molecular scissors.
So we can program a piece of guide RNA to find a specific sequence, say the single T mutation that causes sickle cell disease. The Cas9 enzyme acts as the scissors, physically cutting the DNA at that exact location. And then we can provide the cell with the correct sequence to patch the hole.
The ability to intentionally edit the letters of the manual inside a living organism is a power of unimaginable consequence. It holds the promise of permanently curing genetic diseases at the source, but it also opens profound questions about ecological engineering and the permanent alteration of the human germline.
It is a scientific frontier so massive, with technical mechanics so intricate, that it absolutely demands its own dedicated deep dive in the future. We are just scratching the surface of what active editing means for the future of life on Earth.
I agree completely. Transitioning from readers of the code to authors of the code is the defining scientific narrative of our century.
Well, looking back at what we have uncovered today, it has been a monumental descent into the architecture of life. We started by looking at our own bodies, zooming past the trillions of cells, directly into a microscopic nucleus just a few thousandths of a millimeter wide.
We unravel the impossible 2-meter length of the twisted ladder, freeing it from the magnetic grip of the histone spools.
We examine the physical lock and key physics of hydrogen bonds that dictate the strict pairing of the A, T, G, and C alphabet. We watch the massive mechanical stress of helicase unzipping the strands and polymerase pulling loose chemical letters out of the fluid to build perfect backup copies.
We followed the temporary mRNA blueprints out onto the factory floor, where the ribosome chemically translates those three-letter codons, linking amino acids together until they fold into the dynamic protein machines that perform every action keeping us alive.
And we confronted the paradox of replication. We saw how a physical typo, like a UV photon, fusing thymines together can result in the devastating reality of a misfolded hemoglobin protein, while simultaneously acting as the sole physical engine driving the staggering evolutionary diversity of our planet. It is the ultimate mechanical system. 4 letters, governed by physical thermodynamics, regulated by complex chemical tagging, yielding the entirety of biological existence.
The sheer physical elegance of it is what stays with you.
It is. And as we wrap up, we always like to leave you, our listener, with a final lingering thought. A concept that builds on the mechanics we've discussed today, but pushes our perspective out into the unknown. And our expert has a thought experiment that is guaranteed to shift how you look at the night sky.
It loops back to our very first observation, the profound universality of this molecule. Every single living organism on Earth, from the bacteria in the deep sea vents to the trees in the rainforest, utilizes this exact same sugar phosphate backbone and the exact same A, T, G, and C chemical alphabet. So the provocative question is this: what does the physics of DNA mean for astrobiology?
Right.
If we one day discover life on another world, perhaps swimming in the subsurface oceans of Jupiter's moon Europa, or locked in the ice of Enceladus, when we find that the laws of chemistry and thermodynamics inherently favor this exact same twisted ladder, is a double helix using purines and pyrimidines the most thermodynamically stable way to store data in the universe? Will alien life utilize the exact same 4-letter alphabet, or is DNA just a local earthbound dialect? Will we recognize the code, or will we have to learn to read an entirely alien biology?
Are A, T, G, and C a fundamental rule of the cosmos, or just the way the dice happen to roll on our one tiny rock? I absolutely love that question. It scales the microscopic mechanisms we just discussed to the size of the universe. Thank you for meticulously walking us through the physical mechanics of this molecular manual. And thank you, the listener, for taking this deep dive with us. The next time you scrape your knee or digest a meal or simply look at a mushroom growing on a fallen log, take a second to truly visualize the billions of microscopic twisted ladders unzipping, copying, and folding proteins inside you at this very moment. It is a spectacular mechanical heritage that physically connects us all. We warmly invite you back for our next deep dive into the fascinating source material of our world. Until then, stay curious.